Okay, so welcome. And I'm absolutely delighted to have Professor Sharon DeWitte, evidence of our international pool from the University of South Carolina, who will be giving a talk based on called Social Inequality and Pandemic Mortality in the Black Death. One of the joys of interdisciplinarity, Sharon, I mean, welcome, is that I noticed you were a biological anthropologist, and I had to go away and look up what a biological anthropologist was, and how fascinating I now know. Potted biography. Professor Sharon got a PhD at Penn State in anthropology, was the assistant professor of anthropology, and a member of the human biology program. I'd like to ask you more about that. It's something slightly sinister about that, but I'm sure there isn't. At the University of Albany, and is now a professor at the University of South Carolina. And her work includes the studies of bioarchaeology, which examines the biological, environmental, economic, and social factors that affect and interact with variation in health and mortality, the ecology, epidemiology, and consequences of diseases in past human populations, and the co-evolution of humans and pathogens, which I thought was just really fascinating. And I know it may not be what your talk is about, but I'd kind of want to talk to you about that at the end as well. So welcome, Sharon. Delighted to have you. Over to you.
Thank you so much for having me. This is a real pleasure to be able to share my work with an audience of people from potentially around the world. So I'm going to share my slides and hopefully I will be successful, and we'll start the show. Yes, and how is that looking all right? Okay, great. So again, thank you for having me today. I'm going to talk about sort of the broad sweep of research that I've been doing for about 20 years at this point, and focusing on sort of the ways in which my thinking has evolved since I first started studying the Black Death when I was actually a PhD student. Oops, let me make sure there we go. All right, so the current COVID-19 pandemic is at the very least temporarily focusing the world's attention on the dangers of emerging infectious diseases. Emerging diseases are those that are new to human populations, or that are newly recognized within human populations, or those that are rapidly increasing in their incidence or their geographic range. And dozens of diseases have emerged or have been recognized as emerging over the last few decades, including AIDS, SARS, Ebola, and now COVID-19. And new diseases are certainly going to emerge in the future for a variety of reasons that I'm happy to talk more about for those of you who are interested. So this current pandemic that we're experiencing is caused by a novel pathogen. So that means that it's a pathogen that, as far as we know, is brand new to our species. But this is certainly not the first time that we've experienced a global emerging infectious disease pandemic. And many of the outcomes that we are experiencing currently are not the inevitable result of adding a new pathogen to our populations. And indeed, much of what we have experienced might have been preventable if we hadn't ignored the behavior of and our responses to emerging diseases in the past. So I am a bioarchaeologist, and what that means is that the work that I do relies on data that come from human skeletons that have been excavated from archaeological sites. And generally what I focus on are population level trends in mortality, survival, health, and well-being. At this point, it's been about over 20 years that I have been centering my research around the 14th century Afro-Eurasian pandemic of plague that is now often referred to as the Black Death. This was an emerging disease of the 14th century, and ideally clarifying the context of its emergence and its effects can actually improve our understanding of other emerging diseases, including those that we might face in the future. Molecular evidence now indicates that the pathogen that caused the Black Death was Yersinia pestis, which is the bacterium that causes bubonic plague today. And we also have mounting evidence to indicate that it emerged in Central or East Asia, perhaps as early as the 12th, 13th centuries, and then spread from there across much of Afro-Eurasia, as being indicated in this map produced by Erica Fagan. The Black Death is mostly known for its extraordinarily high mortality levels, although there's recent emerging evidence to indicate that the levels of mortality did vary more than has previously been suspected. But in England, where I do most of my research, it killed an estimated 30 to 60% of affected populations. And that amounted to over 2 million excess deaths within about 2 years, according to Bruce Campbell. And this very high level of mortality in a very, very short period of time brought about, or at the very least hastened, really important demographic, economic, social, and political changes. And then following the Black Death, there were periodic outbreaks of plague for centuries during what we now refer to as the second pandemic of plague. And if anyone wants to know about the first and the third pandemics, I can mention those in the Q&A. In England, where I do most of my research, the Black Death emerged in the context of intense population pressures, increasing social inequality, increasing urbanization, and changing global climatic conditions that were associated with the end of the medieval climate anomaly and the transition to the Little Ice Age. So the prelude to and the experience of the Black Death in England, and specifically in the context of London, where I do a lot of my work, this can potentially provide us with a model for better understanding the confluence of urban environments, inequality, climate change, and emerging infectious diseases. So going back to when I was a graduate student at Penn State, my initial interest in the Black Death was motivated by my broader interest in biosocial variation in health and the phenomenon of selective mortality. So originally, I was really interested in mortality patterns during the Black Death and testing the often made assumption that the Black Death was an indiscriminate killer, that it didn't matter what someone's sex, age, occupation, location of habitation, all of those things, none of those actually mattered in terms of exposure to and death from the Black Death. Now, normal non-epidemic mortality does tend to be selective. And so that means that under normal circumstances, the least healthy or the most frail people in a population are those who are most likely to die. However, many people assume that because of its incredibly high mortality levels, the Black Death behaved indiscriminately and did not exhibit selective mortality. But that assumption had remained largely unexamined until relatively recently. Bioarchaeologists such as myself, we have the potential to test this assumption empirically. And that's because human skeletons can provide data about life, health, and death in the past that are not available in written records, and for people who are typically excluded from written records. So for example, in the context of medieval England, that would include most women, most children, and impoverished people in general. Much of the work that is done in bioarchaeology is informed by eco-social theory, and particularly the idea of embodiment, as described by the social epidemiologist Nancy Krieger. She defined embodiment as how we literally incorporate biologically the material and social world in which we live from utero to death. And a related idea is that no aspect of our biology can be understood in the absence of knowledge of history and individual and societal ways of living. Another core concept of eco-social theory is agency and accountability, which includes a call both to individuals and to institutions to challenge structures and historically and culturally contingent perspectives, attitudes, and behaviors that can potentially harm health. My field of bioarchaeology is also really well positioned, given our attention to biological, social, and environmental contexts, to address the concept of syndemics across time and space. So this concept was introduced by medical anthropologists Meryl Singer and Scott Clare. And what they noted was that diseases are not simply the result of the interaction of a pathogen with a human host's immune system, but rather reflect the synergistic effects of broader individual level and societal conditions. So a syndemic refers to the interaction of two or more diseases or conditions occurring at the same time and the same place, and having health consequences that are greater than simply the sum of their individual effects. At the simplest level, a syndemic can refer to the effects of pre-existing malnutrition on subsequent infection with a pathogen, or pre-existing infection with another pathogen on outcomes of an infection with a subsequent pathogen. So that's basically the simplest way of looking at it. But within anthropology, a syndemic perspective typically acknowledges that the existence and persistence of infectious diseases in our populations and their effects are very deeply intertwined with human sociocultural systems. So the disproportionate incidence and prevalence of diseases among certain populations are very strongly influenced by social factors such as poverty, stigma, racism, sexism, structural violence, and other things. And then ultimately, these directly contribute to the excess burden of disease on these populations, irrespective of the actual causative pathogens of those diseases. So over the years, I've become increasingly motivated to address embodiment and syndemics in the context of the Black Death. And that motivation has only been strengthened by witnessing our failures as a global community with respect to COVID-19 responses. And I'll return to that at the end of my talk. So the reason that bioarchaeologists can examine the Black Death is because during that pandemic, burials were established for the express purpose of accommodating the massive mortality produced by the Black Death. So we have emergency mass burial grounds in many parts of Afro-Eurasia. Most of them have been identified within European context to date. So one of those emergency burial grounds, which I have studied, is the East Smithfield Black Death Cemetery from London. And I'm showing you an image in this slide that was taken during excavation of East Smithfield. It was part of a larger site located near the Tower of London, which you can see in the upper right-hand corner of this image. And this was a site that was used until very recently. And then in the 1980s, there was a redevelopment project that tore buildings down. And in the process of that redevelopment, archaeologists associated with the Museum of London Archaeology Service did the excavation of various levels within, including East Smithfield. So East Smithfield is one of a relatively small number of large excavated cemeteries from Afro-Eurasia that has very strong documentary as well as archaeological evidence linking it specifically to the Black Death, so the inaugural event of the second pandemic. And we know from written records that East Smithfield was established in advance of the Black Death for the sole purpose of burying people who were going to die in the coming plague. So people in England knew that this pandemic was on its way, and they got ready within London to deal with what they knew would be overwhelming deaths. So East Smithfield was used starting in late 1348 or early 1349, and only through the end of the Black Death in London, so the spring of 1350. And what that means is that everybody who's buried in East Smithfield died during the period of the Black Death, and therefore that most, if not all of those individuals were victims of that specific disease. And so it provides a unique opportunity to examine mortality patterns directly by looking at the bodies of people who died during that epidemic. The excavation of East Smithfield yielded over 600 skeletons, many of whom were buried in mass burial trenches that included hundreds of people lined up next to one another and then stacked on top of each other. And other individuals were buried within single graves, so a single person within a grave cut. And all those individuals who were excavated have been curated since then at the Museum of London Center for Human Bioarchaeology. So what I did was I gathered data from nearly 500 people who were buried in East Smithfield, as well as data from nearly 300 people who died under pre-Black Death conditions in the medieval period. So I wanted to get basically a baseline of what mortality patterns looked like prior to the Black Death, to see if and how mortality patterns differed during the Black Death itself. And I was interested in three major patterns of mortality. So the first was whether there were differences in risk of death by sex. And this is important not just for understanding more about the Black Death, but also because we see in living population a lot of variation in susceptibility to and death from diseases by sex. I was also very interested in whether pre-existing health conditions or previous exposure to physiological stressors such as malnutrition or infectious disease, if that had any effect on risk of death during the Black Death. So basically, was the Black Death selective with respect to frailty? And then lastly, I was interested in whether there was variation in risk of death across the lifespan. So in order to assess these patterns, I collected data on age at death for everybody in my sample. So I estimated age at death based on features of the skeleton that change with age. For those of you who are deeply into the literature on bioarchaeology, I used a relatively new method of age estimation for adults called transition analysis. I will spare you those details unless you're really interested. I can talk more about that in the Q&A. But what I do want to emphasize is that I had to rely on estimates from the skeleton for age at death. I don't have any independent information from burial records or headstones or anything else. So this is purely coming from the skeleton. I also estimated sex, and I did that for adult individuals using features of the skeleton that reflect either high levels of estrogen or high levels of testosterone, which in living context, we typically characterize as male versus female, although I would like to emphasize that I recognize that sex is not binary, even though that's often how we treat it for convenience within bioarchaeology. And then lastly, I looked for the presence of a variety of skeletal stress markers. So these are indications on the skeleton that somebody experienced some sort of physiological stressor prior to death. I'm showing three in this slide here. Again, I'm happy to talk more about what each of these might reflect. But what I want to emphasize right now is that none of these skeletal stress markers that I'm showing or any of the others that I included in my research are attributable to the Black Death itself. And I say this because once people exhibited symptoms of the Black Death, which were recognizable at the time, they only lived for another few days, 3 to 5, maybe 7 days, or they recovered. So that's too short a period of time for somebody to manifest a skeletal change in response to a disease process. So these are not attributable to the Black Death itself, but rather give me a general idea of how, what people's experiences of stress were prior to ultimately dying during the Black Death or under normal conditions of mortality. So my general approach in this previous work and in a lot of the work that I continue to do is the application of hazards analysis. And I will show you some slides just to give you a broad idea of how this works. And I will answer any questions anyone has about the specifics of these models. But generally what this approach means is that I'm applying parametric models of mortality to these data that I have estimated from the skeletons. And the benefit of hazards analysis is that these models can accommodate missing data and smooth random variation in mortality data, but they don't impose any particular pattern on the data. So this is what I'm using to look at differences in risk between people with and without stress markers or skeletal lesions. And then these are the models that I use to examine differences by sex as well as variation across the lifespan. And again, if anyone wants to know more about what these parameters are, please ask at the end or ask now if you feel like if you're dying to know what these mean. But what I would like to do instead of getting bogged down in what the parameters are is just cut to the chase and let you know what my main findings were from this previous research. And I just want to highlight that in general, what I'm doing is using a demographic measure to assess health. And this is because health is incredibly hard to define for living people whom you can observe and whom you can interview, for whom you can apply a variety of measures. So it's difficult to assess health for living people. It's even more difficult to assess for bias samples of the dead who are only represented by their skeletal remains. So what I'm trying to do is use demographic measures that are applicable in living contexts to also assess health in the past. And this will come up again later in my talk. So generally what I found were results that indicated that contrary to widely held assumptions, the Black Death was not an indiscriminate killer. And specifically what I found is evidence that older adults faced higher risks of death compared to younger adults, and that across all ages, people who had skeletal signs of exposure to physiological stress experienced higher risks of death during the Black Death compared to their age peers who appeared relatively healthy skeletally. I did not find a strong effect of sex on risk of death during the Black Death. However, more recent historical evidence from the Netherlands does indicate that women faced higher risks of death during the Black Death and subsequent outbreaks of plague, at least in that particular context. So it might be that I did not detect a difference because of the way in which I modeled sex affecting mortality. It might be that my sample sizes weren't sufficient, or there might have been geographic variation in the risks by sex. One of the things that I'm very interested in is how social status and wealth inequality shape risks of death and shape health across time and space. Unfortunately, there is no information about social status for the individuals who are buried in East Smithfield. I will say, though, that some of the variation in health status prior to the Black Death that influenced risks during the epidemic might have been mediated by social status or wealth inequality. And recently, documentary evidence from England and Italy has indicated that in those contexts, in some cases, medieval and early modern plague did disproportionately affect lower status and poor people. So we have mounting evidence that social inequality was having an effect on variation in health and risks of death during the epidemic. So the results that I found that there was variation in risk of death during the Black Death, as well as what we know about its very high mortality levels, raised other questions that I've been trying to answer since then. And one of those questions is, why was the Black Death so incredibly deadly? So I'm showing you in this slide an image of Yersinia pestis, again, the bacterium that we now know caused the Black Death and that continues to cause plague today. Ancient DNA analyses of samples from Black Death victims from East Smithfield and other contexts, it has confirmed that the Black Death was caused by Yersinia pestis, but has not revealed why, perhaps genetically, that strain might have been as deadly as it was. As I mentioned, in England, the Black Death killed 30 to 60% of affected populations. That is a level of mortality that is far beyond anything that has happened in more recent outbreaks of plague, even if you control for antibiotic treatment. So, for example, we know from records of plague outbreaks in India in 1903, 1904, the proportion of the population killed at that point was 2 to 3%, so much lower overall level of mortality. And genomic comparisons of the strain that caused the Black Death to those that circulate today have not revealed anything unique to the 14th century strain of Yersinia pestis that would explain why it was so deadly. And what this means from my perspective is that we actually have to pay very careful attention to the human context, to what was happening in the human population, what factors might have contributed to high mortality levels that are independent of the pathogen itself. So I will note that the skeletal stress markers that I have analyzed can be produced by episodes of malnutrition or infectious disease earlier in life. And I'll also note that because of life history trade-offs, early life stressors can shape immunity and resilience later in life, as predicted by the developmental origins of health and disease model, DOHAD, also referred to as fetal programming. And I'll further note that exposures to stressors can have intergenerational effects via epigenetic changes. Now, a lot of the work that has been done to date on DOHAD and intergenerational effects of stress have focused on non-communicable diseases, as those are the leading causes of death in many populations today. So heart disease, for example. However, there was a study that was published in 2020 by Cheng et al. That found that survivors of the Great Chinese Famine, which lasted from 1958 until 1962, the descendants of those survivors were at increased risk of tuberculosis 2 generations later. So we're starting to get evidence that early life stressors can have intergenerational effects on risks of infectious disease. So putting this all together, this all begs the question to me of what were trends in health prior to the Black Death? Specifically, was the population particularly vulnerable to a new disease because of poor general health resulting from crises that had occurred across multiple generations? And then the other major question that I've been interested in trying to address is, what effect did the selective mortality during the Black Death, perhaps combined with what we know about improvements in standards of living afterwards, what effect did those have on health and demography in the aftermath of the Black Death? So to try to address these questions, I've taken advantage of cemeteries from London that have very good chronological control, at least from a bioarchaeological perspective. And I'm showing you a map here circa 1300 with the locations of the cemeteries that I've studied circled to demonstrate that not only they're all coming from London, but from they're all relatively close to one another. Now, London was incredibly heterogeneous. But at the very least, we know that everyone was coming from the city of London that I'm getting these data from. And so what I was able to do was collect data from nearly 2,000 individuals from these various cemeteries. And I could place each of those individuals into one of 3 time periods. So the earliest is what I refer to as the early pre-Black Death period from 1000 to 1200 Common Era. The second is the late pre-Black Death period from 1200 to 1250, and then the post-Black Death period 1350 to 1540. And what I did was, among other things, and what I'll focus on today, was I assessed temporal trends in survivorship and developmental stress markers. So survivorship is an indication of basically how long on average did people survive in a particular context. And it's another way of talking about life expectancy. And as I mentioned, I use demographic measures to assess health. Survivorship and mortality are 2 demographic measures that we use to assess health in living populations. And specifically with survivorship, in populations that are generally healthy, people can expect to survive relatively long. And in populations that are relatively unhealthy, people experience shorter life expectancies or reduced survivorship. The idea is basically how long are people avoiding the ultimate poor health outcome, which is death. And so I compared trends in survivorship between the early and late pre-Black Death period, and then the late pre-Black Death period and the post-Black Death period. And then I've also, across those time periods, looked at frequencies in a variety of stress markers. But what I'll focus on today is the length of the tibia, which is the larger bone in the lower leg. This I view as a proxy for adult stature. Achieved adult stature can reflect, among other things, exposures to physiological stress during periods of growth. All right, so what I'm showing in this slide are the numerical estimates for mean survival time across those 3 time periods, and then I'll show you graphs in the next slide for those of you who like images. But generally what I found was evidence suggesting that survivorship decreased in the 13th century compared to the 11th and 12th centuries. So in the late pre-Black Death period compared to the early pre-Black Death period, and then survivorship improved following the Black Death. And here are the graphs separated by people estimated to be female and those estimated to be male. The same general trends were I observed for a pooled sample across all sexes and all ages. And what you're seeing in these survival functions is the survival function for the early pre-Black Death period is shown in red or burgundy. And then there's a drop in the late pre-Black Death period. So survivorship decreased. So people were not living as long in the late pre-Black Death period as they had been. And then there was a recovery in survivorship after the Black Death. So people were living longer after the Black Death than they had been prior to in the late pre-Black Death period. Things got really interesting for me when I looked at the results for stature. And what I found was that prior to the Black Death, the stature, well, more technically correct, the tibial length as a proxy for stature for individuals estimated to be male decreased significantly in the late pre-Black Death period. That for estimated females increased slightly, but it was not a significant finding. And then where things get really kind of tricky and interesting is that after the Black Death, the evidence suggests that stature for estimated males increased significantly after the Black Death, whereas stature for estimated females decreased significantly after the Black Death. So what this is suggesting is that there were similar temporal trends in survivorship across sex, but that there were distinct male and female patterns of physiological stress before and after the Black Death. So I'll start with the survival analysis results. What was happening prior to the Black Death? So those pre-Black Death trends were occurring in the context of repeated famines. So in England, in the period from 1000 to 1200, there were 14 famines severe enough to be recorded in contemporary records. And then in the first half of the 13th century, there were 5 famines that were severe enough to be recorded. In England, this pre-Black Death period was also characterized by increasing urbanization, and that would have produced crowded and perhaps unhygienic conditions for many inhabitants of London. This was also a time in England in general of increasing social inequalities. So there was population growth prior to the Black Death that eventually outpaced agricultural production. So by the end of the 13th century, Bruce Campbell has estimated that about 70% of the population of England was living at or below the poverty line. So prior to the Black Death, famine, the interaction between famine and infectious disease, increasing social inequalities in access to food, those all might have exacerbated vulnerabilities to a new disease prior to the emergence of the Black Death. And that might subsequently have made the Black Death more deadly than it might have been if it had struck a population that was in better general health. Now, if you're actually looking at the numbers, you'll note that my skeletal samples do not, they're not from right before the Black Death. So I have skeletal samples up until about 1250 and a little bit after, but mostly 1250. And so you might question whether it's possible that conditions would have improved between 1250 and 1350. Now, I don't have skeletal evidence for that interim, but we do have increasingly good documentary data, mostly from 1270 onward, that suggests that conditions did not improve and in fact might only have deteriorated further prior to the Black Death. So there were, from 1250 on, there were increasingly severe famine conditions associated with climate changes, signaling the end of the medieval warm period and that transition to the Little Ice Age. So for example, the famine that many people are already familiar with, the Great Famine of 1315 to 1317, that killed an estimated 10 to 15% of the population of England. And then that was followed shortly thereafter by the Great Bovine Pestilence of 1319 to 1320, which killed over 60% of bovines in England and Wales, and as a result led to long-term dairy and animal protein deprivations in general in England and Wales, lasting perhaps until the 1330s, so basically at the eve of the Black Death. And these sorts of subsistence crises would have had disproportionate effects on poorer individuals in these contexts. And those poor people were making up an increasing proportion of the population of England. After the Black Death, those improvements in survivorship and by inference improvements in health that were occurring, those were occurring in the context of a severe shortage of laborers that happened because of the mortality during the Black Death. And because of that shortage of laborers in England, wages improved while prices for food and housing and other things were falling. And what happened, at least temporarily, because of these factors is there were improvements in diet and decreases in social inequalities in diet after the Black Death. And that in turn might have led to improvements in general levels of health because diet very strongly determines immune competence and shapes health more generally. There's also evidence that there was an increase in rates of migration after the Black Death, in part because people were no longer tied to their pre-Black Death labor obligations. So the changes that are apparent in the skeletal data after the Black Death, they might reflect a variety of things. So it could be a relatively short-term harvesting effect of the Black Death itself. It might reflect a longer-term selective effect of the epidemic. It might reflect improvements in diet. It might reflect migration of healthy people into the city, or a combination of those things, or others that I haven't considered. So I'm going to turn briefly to the sort of, on the surface, perhaps perplexing results with respect to stature. So what we're seeing for individuals estimated to be males is a congruence of the demographic and stress marker results. And what that might reflect is a relatively straightforward association between developmental stress earlier in life and adult survivorship and health more generally for males during the medieval period. But the results for individuals estimated to be female is less straightforward. So by itself, the apparent significant decrease in adult stature for females after the Black Death might suggest increased physiological stress during childhood and therefore declines in health for females. But at the same time, we're seeing from the skeletal evidence improved survivorship for females. So those are producing two different stories. So what I'm suggesting is that the decline in stature for females might actually reflect improvements in diet or health for females after the epidemic. And I say this because in populations today, improvements in nutrition and reductions in disease burden have been associated with earlier age at menarche, so first menses. And furthermore, in some contemporary populations, there are positive associations between age at menarche and achieved adult stature. And the reason for this is because menarche occurs because there is a surge of estrogen that causes closure of bone growth plates. So first menses is a marker of a cessation of linear growth. Individuals who ovulate and menstruate, once they reach menarche, they only grow a tiny bit after that. So what we see in living populations very often is that a later age at menarche is associated with taller stature, and an earlier age at menarche is associated with shorter stature. So if nutritional status and or disease burdens improve substantially following the Black Death, this might have resulted in an earlier average age at menarche in the post-epidemic population, and therefore an earlier cessation of growth in females and therefore shorter stature after the Black Death. So recently, in an attempt to assess whether this actually might have been happening, I analyzed osteological data on pubertal timing or pubertal stages that had been collected by Dr. Mary Lewis from the University of Reading for the same individuals that I had included in the work that I'm sharing with you today. And what we found is that the average ages of post-menarcheal females or estimated females decreased following the Black Death. And that suggests that there was a decrease in average age at menarche after the Black Death. And that therefore might reflect improvements in living conditions such as diet and disease burdens that occurred after the epidemic. It is also possible, however, that the variation that we're seeing by sex in these patterns might suggest that males benefited disproportionately from improvements in standards of living, including better diets following the Black Death. So altogether, what this suggests is that more work needs to be done. And so that's what I'm attempting to do with my current project in collaboration with Drs. Julia Beaumont and Janet Montgomery. So what we're working on is a project that's integrating multiple lines of evidence to address the mechanisms that were driving those apparent changes in health and demography before and after the Black Death. So we're integrating demographic data from the skeletons, pathological data, isotopic data, and more largely, the work that we're doing will help contextualize another collaborative project that I'm doing that focuses on molecular data reflective of disease ecologies and changes in genetic loci in humans that are associated with immune function and disease susceptibility. I'm also in this project applying intersectionality to examine the non-additive effects of multiple intersecting identities, such as sex, migrant status, and social status, as well as equal social theory, including embodiment that I mentioned earlier, as well as life history theory. So looking at trade-offs across the lifespan related to reproduction and survival. So one aspect of this project incorporates isotopic data reflective of diet, specifically carbon and nitrogen. And what we're interested in is how variation in diet as reflected by these isotopes, what variation was across the medieval period, and specifically if there were improvements in diet as indicated by historical records, were those improvements truly enjoyed by everyone in the population, regardless of age, sex, and social status? And if there were improvements, did those improvements in diet map on to improvements in exposures to stress or resilience to stress? So we're going to be taking or using carbon and nitrogen isotopes and integrating that with information that we have about health and survivorship across the medieval period. And I'm showing you in this slide an image of a tooth that's been cut in half to demonstrate that we're using an approach called incremental dentin analysis that allows us to look at diet during development at a pretty fine-grained resolution. And it specifically will allow us to determine whether people who experience nutritional stress, severe nutritional stress during childhood, what effect that had on later health outcomes in adulthood. And we're also using isotopes reflective of migration or mobility. So we're looking at strontium, oxygen, and lead isotopes to assess temporal trends in migration across the medieval period, and as well as the health patterns of those individuals who are migrating into London. One of the things that we're interested in assessing is the migrant selectivity hypothesis, or the healthy migrant hypothesis, that migrants are a select subpopulation of healthy individuals, versus the possibility that people who migrated into London during the medieval period might have been at higher risk of death from diseases that were endemic within the city that they had not experienced as children elsewhere, or they might have been at higher risk of death because they experienced social marginalization based on their migrant status or other markers of difference, or perhaps because they were impoverished upon arrival and faced the risks that are associated with that status. So far, we have preliminary findings from 93 individuals, and those findings suggest that across the medieval period, including during the Black Death, people who migrated into London suffered lower risks of mortality compared to people who had London isotopic signatures. So it looks like initially we are seeing a signal of that healthy migrant effect. So I'll conclude by hopefully emphasizing the ways in which this work has relevance beyond understanding how people lived and died in the past. Plague is particularly important to study. As I mentioned, the Black Death was caused by the same pathogen, Yersinia pestis, which causes plague today. We have mounting ancient DNA evidence that this single pathogen has infected humans for over 5,000 years and has caused pandemic disease for at least hundreds of years. And it continues to kill people every year. I'm showing you specifically a map of the 2017 plague outbreak in Madagascar, which killed hundreds and hundreds of people. And disturbingly, a very high proportion of cases were the pneumonic form of the disease, which infects the lungs and can be spread directly person to person and is fatal, basically 100% fatal without medical intervention. So plague it's not simply a historic disease. It is a disease that continues to affect people today. It is a zoonosis, so it's a disease of non-human animals, and therefore is impossible to eradicate. And so there's the potential for it to continue to affect people, perhaps devastatingly in the future. So if we can understand better how plague has behaved, how it has affected humans in all of its historical iterations, this is potentially informative about human pathogen coevolution, the effect of climate change on disease emergence and spread, and other phenomena that are really important to get a handle on in order to better deal with diseases in the future. Now, more generally, bioarchaeology provides the human context that is incredibly vital for understanding disease in the past and today. And this is particularly true if we do take a syndemic perspective on disease. So what bioarchaeology can provide is an insight on the interactions among diseases, human biology, human demography, evolution, and social, political, and economic conditions. And what I'm increasingly convinced of is that an important lesson that we might learn from the Black Death is that social inequality prior to the epidemic almost certainly had an effect on health prior to the epidemic, and that might have made the epidemic worse. And the sorts of social inequalities that have been documented for pre-Black Death England and other contexts have been reproduced since then. And we're seeing currently how poverty and social marginalization put people at higher risk of COVID exposure and death from COVID today. Now, these are factors that at least at some point in the lifespan of an individual or in the lifespan of their ancestors are external to the human body. Now, they may become embodied and they might have intergenerational effects because of epigenetic changes, but at least at some point in history, they are external to the human body. So social inequality is not biologically inherent. And if we can show the very deep history of structural conditions that shape human health and that continue to be reproduced today, such as poverty, racism, marginalization of various kinds, that revelation can potentially benefit living people because it can undermine the idea of health as a matter of individual responsibility and can counter any tendency for us to excuse ourselves for not rectifying the structures that create the conditions that are embodied and put people at risk. So I very strongly believe that information about disease in the past can improve responses to disease today. But I don't think I'm alone in being disappointed that we appear to keep making the same mistakes that we made even in the recent past, specifically the 1918 influenza pandemic, where there were also politically motivated resistances to relatively simple but effective measures that can protect public health. So the current pandemic has motivated me to become to be much more explicit in the ways in which I emphasize the deep histories of structural determinants of health in the work that I disseminate to other scholars, as well as work that I publish that will be read by the general public. And when I'm at my most optimistic, I do really think that ancient disease research can be used to harness public interest and raise awareness about the powerful effects that diseases can have on our species. And I think that the Black Death is particularly useful in this respect because so many people are interested in it. So everyone I ever talked to has acknowledged that they know what the Black Death is, and many people are just inherently interested in this one specific historical epidemic. So I think what we can do is leverage that inherent interest to ideally motivate action in the face of epidemics in living populations. And this is ultimately the power of bioarchaeology, providing very richly contextualized information about life in the past, humanizing the past in ways that can potentially benefit people today with respect to these very important topics. And with that, I thank you for your attention, and I'd like to acknowledge people as well as funding agencies that have made this work possible. So again, thank you for having me.